We have studied depolarization, and as depolarization reaches to a sarcolemma muscle fiber, and within muscle fiber, sarcoplasmic reticulum starts releasing calcium, and that calcium ions start coming inside myofibrils, and sarcomere will start performing its function now, and filaments will start sliding over each other. How actually this takes place, we are going to study in detail now within a sarcoma. This is sarcoma that we have studied. This is an outer line. This is called Z lining. This is sarcoma or Z lining. These are the Z lines of the sarcoma. And then there is a region where only actin filaments are present. This is called I band. This is called another I band. And then in between these two I bands, this is actually a region that is called A region, A band. And this A band actually is a band where filaments will slide over each other. And of the A band, there is a region where only myosin is present. To that zone, we say it's called H zone. And the center of the H zone makes a line that's called M line. So I band, another I band, this is called A band, and within A band there is H zone. So for filaments are concerned, these are called actin filaments, and the thick filaments are called myosin filaments, which are made up of myosin molecules. So for this complex is concerned, let's study it first. There are two chains of actins, and these two chains of actins are actually forming a structure of actin filaments. And these filaments are like this. This is one chain of actin, this is called another chain of actin. These two helix-like chains that are drawn in a spherical-like helix-like structure. So the black marker, we would say this is called actin chains. And on this actin, there is another helix-like, double helix-like structure present. This is another filament that's present in a spiral arrangement over actins, and that filament is called tropomyosin. And on these tropomyosins at different intervals, there is another filament present that's called troponin, which actually has three parts. One that binds with the actin, and the other that binds with the tropomyosin, and the other that will bind with the calcium. That will finally bind with the calcium. And this is a structure of troponin. And troponin is present at different intervals on a double helix-like filament, spiral helix-like filament that is called tropomyosin. And tropomyosin is actually a helix-like arrangement that is actually present on an actin, which actually hides the specific sites of actin where myosin hooks can get attached. Therefore, tropomyosin actually is a muscle inhibitor filament. When it hides the sites of actin, myosin heads can't get attached and muscles do not contract. And when muscles are to contract, the tropomyosin just twists and it exposes to the sites of actin, and then myosin filaments can get easily attached and muscle starts contracting. How this all happens, what is going to happen is calcium is released, calcium binds to this structure of troponin. And we have drawn this structure. This is a structure where this actin is surrounded over tropomyosin, and these are sites of actin. And see, these sites of actin are hidden. There is a filament that's called tropomyosin, and tropomyosin is actually hiding to the sites of actin. But as calcium reaches, see in this state, as calcium reaches to binds to a troponin, it actually twists the structure of a tropomyosin, and when tropomyosin is twisted, it exposes the active sites of actin. Now see, this is called this is a tropomyosin which is lifted above, and when it's lifted above, it is exposed site, and the sites of actin where actually the bulbous heads of a myosin can get attached. So what's actually happening? Calcium is released and calcium is binding to troponin, and troponin is actually lifting up. So for example, let me represent it in a way. See, these are three filaments. Consider this one. This is actin, and this blue marker, this center filament is a tropomyosin, and this filament is myosin. When this tropomyosin is present in between them, so the actin sites are not exposed, and these myosin heads can't get attached to this filament. And when you twist it like this, or it's twisted backward like this. Now see, when this is twisted, it can expose to the sites, and these two can now get attached as if there is no filament in between them now, and the sites can get attached easily. So what actually happens at this stage, when calcium binds to troponin, it actually lifts, it actually twists the structure of tropomyosin, and when tropomyosin is twisted, it actually is exposing the sites of actin. And as the sites of actin are exposed, myosin heads can easily get attached. And as they get attached, they will drag actin filaments towards center. And these filaments, these actin filaments will start coming towards center, towards H zone. And as Z lines will start coming near this structure, the length of a sarcomere will reduce, it will become short, and it will become thick, and filaments will start sliding over each other, and that will actually constitute the contraction of muscles. And this would keep taking place. Calcium will continuously keep binding up troponin. It will keep twisting the structure of tropomyosin, that would keep exposing the structure of actin sites, and as actin sites are exposed, myosin bulbous heads can get attached, and they can drag them towards center, and this is how a whole sarcomere will start contracting, and this actually is a mechanism of a muscle contraction. And in that contraction, there is a role of energy also, and where actually energy is required. See these heads. They, in order to get attached to the active sites, they consume one molecule of ATP. See, one molecule of ATP is consumed, and a bulbous head is lifted up, and when it's lifted up, it gets attached to the sites of actin. And it not only gets attached, but it bends and it drags actin filaments towards center. And once it's dragged, it again needs to detach and to rebind with the actin sites again. And in order to detach again, it again consumes another molecule of ATP. That consumes another molecule of ATP and it detaches back. And the cycle keeps continuing. It becomes flat again, and it again needs ATP to uplift and to get attached to active site and to contract and to move actin filaments towards H zone, towards center. And once it's dragged, it again needs ATP to detach. And this is a continuous structure that myosin bulbous heads keep repeating, and for that, for attachment and for detachment, they are both time require two molecules of ATP. Therefore, there is such a huge amount of ATP required for the contraction of muscles. We are going to study where from this ATP comes later. They can just understand what actually happens. Calcium binds to troponin. It actually lifts to the structure. Say for example, if this is tropomyosin, it simply lifts the tropomyosin in its position. It actually exposes the sites of actin, and when actin sites are exposed, another filament, we can say, myosin heads can get attached and muscle starts contracting. So this is a simple mechanism of contraction of muscles where actin sites will continuously be attaching to the hooks, to the cross bridges of myosin, and that would be, myosin would be dragging them towards center, and gradually H zone would start decreasing, and A band, and then the filaments start overlapping in a A band region. And in this way, gradual decrease in H zone will bring these Z lines together, and sarcomere would be reduced and it will become short, and that would constitute the contraction of muscles. And for that, as I said, energy is required, and where from that energy comes, muscles require energy, and that energy is coming from glucose, it comes from glycogen, it comes from fatty acids, it comes from proteins. Energy is actually provided from respiration. Most of the food is respired, and the oxidation of food actually produces ATP, and that ATP is consumed for energy consuming functions, your contraction of muscles. There is another source of energy also, that is called keratin phosphate. And keratin phosphate is a proteinous product that is present inside sarcoplasm, and keratin phosphate can break into release a phosphate that can combine to ADP, and it can instantly synthesize ATP, which can also be available for contraction of muscles. So this is another source of ATP that is also present in sarcoplasm to help in a contraction of muscle. And when we say energy comes from respiration, respiration can be of two types. It can take place aerobically and it can take place anaerobically. And when there is a sufficient amount of oxygen present, muscles respire aerobically and they break glucose first into two molecules of pyruvate by a process of glycolysis. And later these two molecules are further broken down by a process called Krebs cycle, where pyruvates break down into carbon dioxide and water. And during Krebs cycle, substrate level phosphorylation is taking place, but actually it synthesizes different kinds of carriers called NADH and FADH. And all these NADH and FADH finally oxidize and they give electrons to a chain that is present in the crystal of mitochondria, that's called electron transport chain. And finally, these electrons are received by oxygen and it takes protons and it synthesizes water. And in this way, oxygen is finally required to complete the process of respiration. Therefore, we say it is called cellular respiration, where glycolysis produces ATP, Krebs cycle produces ATP, and sufficient amount of ATP is produced by oxidative phosphorylation, which is going to take place through electron transport chain, where oxygen is actually helping to settle all these electrons finally. Therefore, this is said to be a respiration called aerobic respirations. There can be another respiration that's called anaerobic respiration. And in anaerobic respiration, glucose definitely first breaks into pyruvate. That's a common process both in aerobic and anaerobic. And after glycolysis, the pyruvate cannot completely break down because there is an absence of oxygen. And when oxygen is not available, pyruvate doesn't break into carbon dioxide and water, but it converts into lactic acid. And when pyruvate is converted into lactic acid, it's not completely broken down. Therefore, it's not releasing sufficient amount of energy. Therefore, we can say this anaerobic respiration produces no amount of ATP. On the contrary, lactic acid itself is a toxic. And when this toxic is stored in the muscles, it produces a tiring effect in the muscles. Therefore, we say that causes a problem that's called muscle fatigue. So this is another story that relates to the energy for muscle contraction. You must be knowing that when we are performing a heavy exercise, where the oxygen that we have taken is consumed almost, and we are running shortage of oxygen, and muscles are not provided with a sufficient amount of oxygen, still we are performing activity, and for that we are requiring energy, and muscles, in order to meet the requirement of energy, start respiring anaerobically. And when they start respiring anaerobically, they just keep on performing that function by energy that's coming out of glycolysis. Because they do not have another source of energy, they will have to rely on a process of anaerobic respiration, and their anaerobic later is not producing ATP, just a process of glycolysis would keep on producing ATP, which would be though little, but at least it is better than something, it's something better than nothing. So glycolysis would keep providing energy, and the pyruvates later, as there is no oxygen, would change into a lactic acid that would resultantly lead to accumulation of lactic acid, and that would finally cause muscle fatigue. And definitely we will have to stop that activity, exercise or functions when our muscles will finally stop performing functions, as they would be tired enough because of the accumulation of lactic acid, which we call it's called muscle fatigue. So this is actually a process of a muscle contraction, where it's nerve impulse we have studied. We have studied how calcium releases, and we have studied where calcium binds, and where sarcomere and filaments twist, and how these filaments get attached with each other and they contract. And for that, attachment and detachment of myosin aids energy, and where from that energy comes, this is how is the story of the contraction of muscles.